Hi, I'm Karen Stever, and welcome to Biostimulants Part 3 out of 6. In this episode, I'll talk about humates and chitosan. There are two types of molecules derived from natural products. Here's a list of bullet points describing the effects of biostimulants on plants. I've probably beaten the top of this list pretty much to death, but down here at the bottom, I started introducing things in the last episode, in particular how plant genes can be turned off and on and how metabolic pathways and the levels of small molecules produce change. As we go through the next sections, we're going to be adding more data to demonstrate these concepts. Starting with humates or humate substances, these are large molecule substances derived by fractionation of leonardite, a type of coal. That fractionation is pretty similar to the process of oil refining into gas and other products. From this fractionation, we get low molecular weight component, the fulvic acids, and fulvate if it's in the basic form. Higher molecular weight is humic acid or humates. It's still soluble, but it is higher molecular weight. And then the highest molecular weight humates will not be soluble. So we're going to be dealing in horticulture mostly with fulvic and humic acid. Those can be formulated into both liquid and solid formulations. We also get humates from the humus that's in compost tea, and worm castings. So again, aged organics, but not quite as aged as coal. So the humic substances are well established to have a beneficial effect on plants. Here I want to present some data where the researchers looked to see exactly what was going on in the cells. They were looking for the changes in the expression of genes, so production of proteins, enzymes in the cells that they could monitor and follow. In this display, which is called a heat map, the orange coloration means that the level of those in protein products did not really change if it's orange. If it's gone to more yellow, their production has been decreased or their level in the cells is decreased, and if it goes to more red, the level in the cells has increased. So my description completely glosses over the complexity of these experiments, but what I want to say is it's clear that many of the genes were initially downregulated at 24 hours, but then were increased, their expression was increased, they were upregulated by 72 hours. What the small molecule increases accompanied by these changes in the protein were an increase in jasmonic acid, IAA, the rooting hormone, and abscisic acid. So those cellular changes that occur with the addition of humates or humic substances, that was all happening inside the cell. But outside, you're going to be able to see the additional root growth, sometimes additional chlorophyll content, stress tolerance when the plants are challenged, can measure increases in respiration and in the nutrient uptake of plants easily, and also the soil itself is changed with humic acid. This cation binding capacity is increased due to the capacity of the humates. So those are all beneficial effects that occur with the humates. Next, I'm going to switch to chitosan. Chitosan is derived from another natural product, this time the chitin in crustacean shells, mushrooms, and some other fungi. Chitin is shown here at the bottom. That's our source material. It's comparable to cellulose in this polysaccharide chain. In order to convert the chitin to chitosan, it's subjected to acid or base and deacylation, so we're removing these two carbon chains here off of that nitrogen. Once that's done, and it can be done to different levels, so chitosans will vary in their activity, their solubility, and other things by the amount of this deacylation, by the source of the original starting material, and also by the exact nature of the acid and base. So chitosan is not one product, it's kind of a range of products, let's say. But they are soluble and can be applied as the drench or the spray, and chitosan does have good biostimulant effect. So I'm going to show an experiment here to demonstrate the power of chitosan. In this experiment, here's our control plant on the left. The second plant has had a growth inhibitor added to it. If we add chitosan with that growth inhibitor, we've reversed all of the inhibition. If we add chitosan to a plant that's never seen that inhibitor, we get a much beefier plant than the original control. So the benefits of chitosan are improved growth, vigor, more fruit, more flowers, higher yield, increased chlorophyll, more photosynthesis, and increased chemical content, so upping the small molecules of the plant. The chitosan does induce reportedly systemic acquired resistance. That is different. That is a whole plant effect. It's a stronger effect than the ISR pathways we've been talking about. Chitosan directly reduces fungal, bacterial, and viral pathogens, and even nematodes. So it can be really beneficial to your crops in a number of ways, not just with the induced resistance pathways. Here's another trial of chitosan on strawberries where it was trialed from 125 parts per million up to 1,000. The red arrows indicate here where they got the best fruit yield out of this crop. That was at 125 to 250 parts per million. Now, what I will say, that is specific to the formulation they were using, because there are quite a few variables in chitosan as there are in some of these other natural products. The first is the source material, but then in the processing, the degree of deacylation or polymerization will affect the overall properties of the product. And the other technicality is that we don't have a major horticultural supplier producing this. So other smaller players are entering the market and bringing chitosan into the domain, but I would say it still has a way to go. So if you want to try some of these biostimulants on your crops, and I hope that you do, I want to recommend that you trial them side by side, treated and untreated, so that you can really see what these biostimulants are doing for you. With the products I talked about today, or the biostimulants, more is not better. It is possible to overdose both the chitosan and the humates. It's probably not easy. So stay inside the recommended rate ranges by your manufacturer, but if that range is wide, trial a variety of rates within the range. So thanks for joining in today. I hope I have given you a little bit of information that you can use. If you should have any questions, comments, or concerns, please call or email me at the contact information seen here. And be sure to tune in for our next episodes, especially episode 6. I think you'll find it interesting.